Welcome to my channel. Today, we're diving into one of the most fascinating and complex organs in the human body, the brain. Our journey begins with a look at the brain from a lateral view, or a side view, which provides a unique perspective on its major structures. This view allows us to appreciate the intricate design of the brain, from the folds and grooves on the surface to the deeper areas that control essential functions like movement, memory, and emotions. Whether you're interested in understanding how we think, learn, or feel, this exploration of the brain will offer insight into the very essence of what makes us human. Let's start by identifying some key regions visible from the side. As we examine the brain from this lateral view, the first thing you'll notice is its outermost layer, the cerebral cortex. This wrinkled surface is where much of our higher cognitive functions, like decision-making and problem-solving, take place. But there's so much more hidden beneath these folds. One of the most noticeable features from this angle is the frontal lobe, located at the front of the brain. This area is crucial for controlling voluntary movement, speech, and our ability to plan and organize. It's also heavily involved in personality and emotional regulation. Moving towards the middle of the brain, we encounter the parietal lobe, which plays a key role in processing sensory information, everything from touch to spatial awareness. If you've ever reached for something without looking, the parietal lobe is helping guide your movements. Just behind that is the occipital lobe, found at the back of the brain. This region is dedicated to vision. Whenever you look at an object, your occipital lobe is working to process the images your eyes send to your brain. Next, below the parietal lobe, we find the temporal lobe. This area is involved in hearing, memory, and understanding language. It's also closely tied to our ability to recognize faces and emotions, making it a critical player in social interactions. Lastly, tucked under the occipital lobe and extending down from the brainstem, we have the cerebellum. Although it's smaller in size, the cerebellum is crucial for balance, coordination, and fine motor skills. Whenever you move smoothly or keep your balance, thank your cerebellum. Now that we've identified the major lobes from the lateral view, let's take a closer look at what each of these regions does and why they're so important to everyday life. Starting again with the frontal lobe, this area is sometimes referred to as the control center of the brain. It's where we engage in critical thinking, problem-solving, and decision-making. The frontal lobe also houses the motor cortex, a strip of tissue that sends signals to your muscles to initiate movement. When you wave hello or walk across a room, your motor cortex is hard at work. Additionally, the prefrontal cortex, the front part of the frontal lobe, is key to personality development and social behavior. It's the reason why we can control impulses and plan ahead, giving humans a higher level of cognitive flexibility compared to most animals. Next, let's explore the parietal lobe. This lobe integrates information from your senses, helping you understand and interact with the world around you. The somatosensory cortex, located here, is responsible for processing sensations like touch, temperature, and pain. When you touch something hot or feel the texture of an object, this part of your brain interprets that sensory information. It also plays a role in spatial orientation, helping you recognize where your body is in relation to objects and your environment. The occipital lobe, as we mentioned earlier, is the brain's visual processing hub. Everything you see is processed here before being interpreted by other parts of the brain. Damage to the occipital lobe can lead to visual impairments or even blindness, despite the eyes functioning normally. It's a reminder of how interconnected our brain and senses really are. Moving forward, the temporal lobe, this is where your brain processes sounds and language. The auditory cortex located here helps interpret the sounds you hear, whether it's music, speech, or background noise. Another critical area in the temporal lobe is the hippocampus, which plays a major role in forming new memories. The hippocampus helps us store and retrieve information, whether it's remembering where you left your keys or recalling an important life event. In addition, the temporal lobe is vital for understanding spoken language and is closely associated with emotional regulation, thanks to its connection with the amygdala. Lastly, let's revisit the cerebellum. Often called the little brain, this structure coordinates fine motor skills. It works like an internal gyroscope, helping you maintain balance and posture. The cerebellum is constantly adjusting your movements to keep them smooth and controlled, whether you're riding a bike or playing a musical instrument. Though it's not as big as the cerebral cortex, the cerebellum is crucial for the precision and timing of your actions. To truly understand how the brain functions, it's important to look at how it develops, both in the individual and through evolutionary history. The brain is often described as having three major parts that reflect its evolutionary development: the hindbrain, the midbrain, and the forebrain. These regions can be seen in a developing fetus and are also part of what makes up the brains of other species, showing how humans have adapted over time. First, let's start with the hindbrain, which includes structures like the medulla oblongata, pons, and cerebellum. These areas are responsible for the most basic functions necessary for survival, like breathing, heart rate, and coordination. Evolutionarily, this is one of the oldest parts of the brain, shared with many animals. It develops early in fetal growth and is essential from the moment a baby is born, controlling things we don't have to think about, like breathing and digestion. Next is the midbrain, which includes the mesencephalon. This structure is crucial for coordinating movement and sensory information. The mesencephalon is also involved in functions like vision, hearing, and motor control. It contains regions like the superior colliculus, which helps orient the eyes and head towards stimuli, and the inferior colliculus, which plays a key role in processing auditory information. The midbrain connects the higher-level thinking of the forebrain with the basic functions of the hindbrain, acting as a bridge between these two systems. Lastly, we have the forebrain, the most advanced and evolutionarily recent part of the brain. This region includes the cerebral cortex, which we discussed earlier, along with deeper structures like the thalamus, hypothalamus, and basal ganglia. These areas are involved in complex functions like decision-making, emotional regulation, and sensory integration. The forebrain is what sets humans apart from other animals, giving us advanced cognitive abilities and a higher level of consciousness. Mesencephalon, although small, the mesencephalon plays an essential role in our brain's overall functioning. It contains two key areas, the tectum and the tegmentum. The tectum handles visual and auditory reflexes. If you've ever quickly turned your head towards a loud noise or a sudden flash of light, the tectum was responsible for that reflex. The tegmentum, on the other hand, is involved in controlling motor functions, regulating awareness and attention, and handling some aspects of pain perception. The substantia nigra, a part of the tegmentum, produces dopamine, a neurotransmitter important for movement and reward. Damage to the substantia nigra is linked to Parkinson's disease, which highlights how crucial these midbrain structures are for motor control. As a fetus develops, the brain undergoes rapid changes, starting as a simple neural tube. By the 5th week of gestation, the brain begins to divide into its primary sections, the forebrain, midbrain, and hindbrain. These regions continue to grow and specialize throughout pregnancy, becoming the complex organ that controls everything from automatic functions to conscious thought. The mesencephalon, in particular, plays a key role early in development by forming connections between the spinal cord and higher brain centers. This helps prepare the developing brain to coordinate basic sensory and motor functions before more complex structures like the cerebral cortex mature. Over time, the brain builds upon these lower, more primitive regions to support advanced cognitive functions, but it never loses its dependence on the mesencephalon and other core structures. By birth, the brain has developed most of its basic structures, but it continues to grow and refine neural connections throughout childhood and adolescence. In fact, the brain doesn't fully mature until the mid-20s, with areas like the prefrontal cortex, the center for reasoning and decision-making, being the last to fully develop. The limbic system is a group of interconnected structures deep within the brain that play a crucial role in processing emotions, forming memories, and driving behaviors essential for survival. Often referred to as the emotional brain, the limbic system helps regulate our responses to stimuli, especially those related to fear, pleasure, and motivation. Key structures of the limbic system. Let's explore the major components of the limbic system and their specific functions. Amygdala. The amygdala is a small, almond-shaped cluster of nuclei located deep within the temporal lobes. It is often described as the brain's fear center, but its role extends to other emotional responses, such as anger, pleasure, and anxiety. Functions. Emotional processing. The amygdala is critical for recognizing and responding to emotionally significant events, especially threats. It generates quick, instinctual responses, such as the fight or flight reaction. Fear and aggression. It helps in processing fear and aggression, contributing to the formation of fear-related memories. For example, when faced with danger, the amygdala activates the body's stress response by stimulating the hypothalamus and releasing stress hormones. Emotional learning. The amygdala is also involved in forming emotional memories, associating certain stimuli with emotional experiences, e.g., a person associating a particular location with fear after a traumatic event. Fun fact, in some experiments, Animals with damaged amygdalas show a lack of fear or inability to recognize danger, which highlights its crucial role in survival. Hippocampus. The hippocampus is a seahorse-shaped structure located in the temporal lobe, just behind the amygdala. It is best known for its vital role in memory formation and spatial navigation. Functions: memory formation. The hippocampus helps convert short-term memories into long-term ones, especially episodic memories and declarative memories. Damage to the hippocampus can lead to difficulties forming new memories, as seen in conditions like anterograde amnesia. Spatial memory. The hippocampus also plays a role in understanding spatial relationships and navigation, such as remembering directions or locations. Memory recall. During memory recall, the hippocampus helps retrieve previously stored memories by activating relevant areas of the brain. Fun fact: the hippocampus is one of the few areas in the brain capable of neurogenesis, meaning it can grow new neurons throughout life, which is thought to be important for learning and memory. Hypothalamus. The hypothalamus is a small but vital structure located below the thalamus and just above the brainstem. It acts as a control center for maintaining the body's internal balance, known as homeostasis. It also plays a critical role in regulating emotions, hormone release, and bodily drives like hunger and thirst. Functions: emotion and stress response. The hypothalamus regulates the body's response to emotional stimuli by controlling the release of hormones from the pituitary gland, such as cortisol during stress. Homeostasis. It controls body temperature, hunger, thirst, sleep, and circadian rhythms. When you're thirsty or hungry, it's the hypothalamus signaling these needs. Sexual behavior and motivation. The hypothalamus regulates sexual arousal and reproductive behaviors by releasing hormones like oxytocin and testosterone. Fun fact: the hypothalamus is responsible for triggering fight or flight responses during stress by activating the sympathetic nervous system, but it also promotes rest and digest activities when the body is relaxed. Thalamus. The thalamus is often called the brain's relay station because it acts as a hub for sensory information, directing it to the appropriate regions of the cortex for further processing. Functions: sensory processing. The thalamus receives sensory information and sends it to different parts of the brain for interpretation. For example, visual signals from the eyes go to the visual cortex via the thalamus. Emotion regulation. While not primarily involved in emotion, the thalamus helps filter incoming sensory information that could trigger emotional responses, sending important stimuli to other areas like the amygdala and cortex for further evaluation. Fun fact: the thalamus plays a role in sleep and wakefulness, helping regulate arousal and consciousness, which are essential for maintaining attention and awareness. Cingulate gyrus. The cingulate gyrus is part of the limbic cortex and is involved in regulating emotional responses and linking them to behavioral outcomes. It is crucial for empathy, decision-making, and self-control. Functions: emotional regulation. The cingulate gyrus helps to regulate emotions by integrating emotional information and influencing emotional responses. It plays a role in empathy and emotional expression. Behavioral regulation. This structure is key in the process of making decisions based on emotional input, which can influence how we approach situations. Fun fact: the cingulate gyrus is often more active when people are in conflict or need to exert self-control, such as resisting temptation. The limbic system's role in emotion and behavior. The limbic system is crucial for survival-oriented behaviors and emotional responses, including fear and aggression. The amygdala and hypothalamus help trigger fight or flight responses in threatening situations, while other parts of the limbic system regulate the stress response. Pleasure and reward. The limbic system is key to understanding pleasure and reward. The nucleus accumbens, though not traditionally part of the limbic system, works closely with it to process feelings of reward and motivation, often mediated by dopamine. Motivation and drives. Limbic structures influence fundamental drives like hunger, thirst, and sexual behavior, connecting basic bodily needs to emotional responses. Limbic system and memory. Memory and emotion are deeply intertwined in the limbic system. Emotionally charged memories, memories with a strong emotional component, whether positive or negative, are often more vivid and easier to recall because of the involvement of the amygdala and hippocampus. Trauma and fear memories. The amygdala's role in fear processing explains why traumatic events often leave such lasting, powerful memories. These memories can be protective but, in some cases, lead to conditions like post-traumatic stress disorder. Clinical relevance. Limbic system disorders. The limbic system can be implicated in various mental health and neurological conditions, such as anxiety and depression. Dysregulation of limbic structures, particularly the amygdala and hippocampus, has been linked to mood disorders. Overactivity in the amygdala can result in heightened anxiety, while a shrunken hippocampus is often observed in individuals with chronic depression. Alzheimer's disease. The hippocampus is one of the first areas affected by Alzheimer's, which is why memory loss is an early symptom of the disease. Addiction. The limbic system, particularly the reward pathways involving the nucleus accumbens, is involved in the formation of addictive behaviors, reinforcing behaviors like drug use or overeating by creating feelings of pleasure. Summary of the limbic system. In summary, the limbic system is a network of brain structures that orchestrate our emotional responses, memory formation, and basic survival instincts. Its key structures include amygdala, fear, aggression, and emotional memory. Hippocampus, memory formation and spatial navigation. Hypothalamus, homeostasis, emotion, and hormonal regulation. Thalamus, sensory relay and emotion filtering. Cingulate gyrus, emotional regulation, empathy, and decision-making. Together, these structures form the emotional core of the brain, influencing not only how we react to the world but also how we remember it and act upon it. When examining the brain, it's crucial to understand the distinction between white matter and gray matter, two types of tissue that make up the brain and spinal cord. Though they may sound similar, they serve very different roles in how the brain functions. Gray matter. Gray matter consists mainly of neuronal cell bodies, dendrites, and unmyelinated axons. It's where the actual processing in the brain happens, involving everything from muscle control to sensory perception. When we talk about the cerebral cortex, we're largely referring to gray matter, the outermost layer of the brain that handles higher-level thinking, memory, and decision-making. Gray matter also extends deep into the brain, forming areas like the basal ganglia, which help regulate movement, and the thalamus, which relays sensory signals to the cortex. Since it processes information, gray matter is abundant in regions involved in cognition, emotion, and movement. White matter, on the other hand, consists mostly of myelinated axons, long nerve fibers coated in myelin, a fatty substance that helps transmit signals efficiently. While gray matter processes information, white matter serves as the brain's communication network, connecting different regions and allowing for the fast transmission of information between them. The myelin sheath gives white matter its color and speeds up the electrical signals between neurons. This enables different parts of the brain to coordinate activities quickly, whether it's sending a motor command or retrieving a memory. White matter makes up the deeper layers of the brain and can be found in large tracts like the corpus callosum, which connects the two hemispheres of the brain. Interesting facts: the human brain contains about 86 billion neurons, with gray matter making up about 40% of the brain's mass, while white matter comprises the remaining 60%. Gray matter density can increase with mental exercises and learning, showing that brain plasticity is closely tied to this tissue. White matter deteriorates with age, but maintaining mental and physical activity has been shown to slow down this process, supporting cognitive function later in life. The corpus callosum, the largest white matter structure, contains over 200 million axons, facilitating communication between the brain's left and right hemispheres. Next, let's look at the brain's sulci and gyri. These are the ridges and grooves that give the brain its characteristic wrinkled appearance. They may seem like just surface features, but they play a critical role in how the brain is organized and how much information it can process. Sulci. A sulcus is a groove or depression on the surface of the brain. These grooves help divide the brain into different regions or lobes and allow for greater surface area without increasing the brain's overall size. The more sulci there are, the more space there is for neurons, enabling the brain to process more information. Some notable sulci include: central sulcus, separates the frontal lobe from the parietal lobe. It also divides the primary motor cortex from the primary somatosensory cortex, which is essential for voluntary movement and sensory processing, respectively. Lateral sulcus, separates the frontal and parietal lobes from the temporal lobe below. This sulcus is important for locating the regions of the brain involved in hearing and speech comprehension. Gyri. A gyrus is a ridge or fold between two sulci. These folds increase the brain's surface area, allowing for more neurons to fit into the same space. Each gyrus has specific functions based on its location. Some key gyri include: precentral gyrus, located in the frontal lobe. This ridge contains the primary motor cortex, which is responsible for controlling voluntary muscle movements. Postcentral gyrus. This ridge lies just behind the central sulcus in the parietal lobe and houses the primary somatosensory cortex, which processes touch, temperature, and pain sensations from different parts of the body. Superior temporal gyrus, located in the temporal lobe. this gyrus is important for processing sounds and comprehending spoken language. Damage to this area can lead to difficulties in understanding language, known as Wernicke's aphasia. Why are the sulci and gyri important? The grooves and ridges maximize the brain's surface area, allowing for more neurons and, consequently, more computational power. This is essential for the brain's ability to perform complex functions like abstract thinking, problem solving, and language processing. Interestingly, the pattern of sulci and gyri is unique to each individual, much like a fingerprint, although certain key sulci and gyri are common across all human brains. Let's now shift our focus to neuron structure and the various types of cells in the brain. This will provide insight into how the brain processes information and supports its functions on a cellular level. The brain's ability to process and transmit information relies on neurons, the basic building blocks of the nervous system. Each neuron is a specialized cell that communicates with others through electrical and chemical signals. Although neurons come in various shapes and sizes, they all share common features that allow them to function effectively. Cell body. This is the core of the neuron where the nucleus is located. The cell body contains the neuron's genetic material and is responsible for maintaining the cell's overall health. It's also where proteins are synthesized, which are essential for sending signals to other neurons. Dendrites. Dendrites are branch-like extensions that project from the cell body. Their primary role is to receive signals from other neurons. Think of dendrites as the input channels for the neuron. They collect information in the form of neurotransmitters from neighboring neurons and pass it along to the cell body for processing. Axon. The axon is a long, slender projection that extends from the cell body. It functions as the output channel, transmitting electrical impulses away from the neuron to communicate with other neurons, muscles, or glands. Some axons are incredibly long, such as those in the spinal cord, which can extend all the way down the body. Myelin sheath. Surrounding the axon in many neurons is the myelin sheath, a fatty insulating layer that helps speed up the transmission of electrical signals. This insulation is critical for efficient communication between neurons. The gaps in the myelin sheath, known as nodes of Ranvier, allow the electrical signal to jump quickly along the axon, increasing the speed of transmission. Axon terminals. These are the endpoints of the axon, where the neuron connects to other cells. The axon terminals release chemicals called neurotransmitters into the synapse, a small gap between neurons. This is how one neuron sends signals to the next. These neurotransmitters bind to receptors on the neighboring neuron's dendrites, thus passing along the message. Interesting facts about neurons. A human brain contains around 86 billion neurons. Each neuron can have thousands of synapses, forming a vast and intricate communication network. It's estimated that there are 100 trillion synaptic connections in the human brain. Neurons don't regenerate in most parts of the brain, but some regions, like the hippocampus, can produce new neurons throughout life through a process called neurogenesis. Types of cells in the brain. In addition to neurons, the brain contains several other types of cells that play critical roles in maintaining its function. These cells are collectively known as glial cells, and they serve to support and protect neurons. Let's explore the main types of brain cells. Astrocytes. Astrocytes are star-shaped cells that provide structural and metabolic support to neurons. They help maintain the blood-brain barrier, which protects the brain from harmful substances in the bloodstream while allowing necessary nutrients to pass through. Astrocytes also regulate the chemical environment around neurons by controlling the levels of neurotransmitters and ions. Functions: provide nutrients and oxygen to neurons. Maintain the balance of chemicals like potassium and neurotransmitters. Aid in the repair and scarring process following injury in the brain. Fun fact: astrocytes play a role in synaptic plasticity, meaning they influence how neurons strengthen or weaken their connections based on activity, which is essential for learning and memory. Oligodendrocytes. Oligodendrocytes are responsible for producing the myelin sheath that insulates the axons of neurons in the central nervous system. By wrapping around multiple axons, oligodendrocytes allow faster transmission of electrical signals along neurons. Functions: form the myelin sheath around axons in the CNS. Speed up the transmission of nerve impulses. Fun fact: in the peripheral nervous system, a different type of cell called Schwann cells performs the same function as oligodendrocytes, wrapping axons in myelin. Microglia. Microglia act as the immune cells of the brain. They protect the brain by constantly surveying for signs of damage, infection, or inflammation. When they detect trouble, microglia engulf and digest debris, dead cells, and foreign invaders. Functions: act as macrophages that clean up damaged cells and pathogens. Play a role in the brain's inflammatory response. Help clear out unnecessary synaptic connections during brain development and learning. Fun fact: microglia are involved in what's known as synaptic pruning, a process that removes weaker synaptic connections and strengthens others, helping to refine neural networks, especially during early brain development. Ependymal cells: these cells line the ventricles of the brain and the central canal of the spinal cord. They produce and help circulate cerebrospinal fluid, which cushions the brain and spinal cord and provides them with essential nutrients. Functions: produce and regulate cerebrospinal fluid. Act as a barrier between the CSF and nervous tissue. Fun fact: the CSF produced by ependymal cells acts as a shock absorber, protecting the brain from injury due to sudden movements or impacts. Radial glial cells: these cells are critical during brain development as they guide the migration of neurons to their appropriate positions in the brain. Though most disappear after development, some radial glial cells persist and become astrocytes or other glial cells. Functions: guide developing neurons to their final positions in the brain. Play a role in neurogenesis in certain brain regions. Summary of neuron and glial cell interactions: the interaction between neurons and glial cells forms the backbone of the brain's functionality. Neurons are the primary processors of information, using electrical impulses and neurotransmitters to communicate with each other. Meanwhile, glial cells provide critical support, insulation, immune defense, and nutrient supply to ensure that neurons can function efficiently. While neurons often take the spotlight, it's important to remember that without the help of glial cells, like astrocytes maintaining the chemical balance or oligodendrocytes speeding up communication, the brain wouldn't be able to perform even its most basic tasks. What are neurotransmitters? Neurotransmitters are chemical messengers that neurons use to communicate with each other, muscles, or glands. When an electrical signal, called an action potential, reaches the end of a neuron, it triggers the release of neurotransmitters into the synapse, the small gap between neurons. These chemicals then bind to receptors on the neighboring neuron's dendrites, transmitting the signal and allowing the neurons to communicate. Neurotransmitters play a key role in regulating various physical and psychological functions, including mood, memory, movement, and heart rate. Once released, neurotransmitters can have different effects, either exciting or inhibiting the neighboring neuron. Excitatory neurotransmitters increase the likelihood that the receiving neuron will fire an action potential, while inhibitory neurotransmitters decrease this likelihood, essentially calming the brain's activity. Key neurotransmitters and their functions. Here are some of the most important neurotransmitters in the brain, along with their roles and functions. Dopamine. Function. Dopamine is a neurotransmitter associated with reward, motivation, and movement control. It plays a crucial role in the brain's reward system, reinforcing behaviors by providing feelings of pleasure when we experience something enjoyable, like eating or achieving a goal. It also helps regulate movement by coordinating motor control pathways. Roles. Reward and pleasure. Dopamine is released when we engage in rewarding activities, creating a sense of pleasure and reinforcing those behaviors. Motivation. Dopamine drives goal-directed behaviors and motivates us to pursue rewards. Motor control. Dopamine-producing neurons in the substantia nigra are vital for controlling movement. A deficiency of dopamine in this region is associated with Parkinson's disease. Fun fact: dopamine is often called the feel-good neurotransmitter because it's closely linked to the brain's reward system. However, it's more about motivation and desire than actual happiness. Serotonin. Function. Serotonin is a neurotransmitter that plays a major role in mood regulation, sleep, appetite, and digestion. It is often referred to as the mood stabilizer because it helps promote feelings of well-being and emotional stability. Roles. Mood. Low levels of serotonin are associated with depression and anxiety. Many antidepressant medications, like SSRIs, work by increasing the availability of serotonin in the brain. Sleep. Serotonin is involved in regulating the sleep-wake cycle and is a precursor to melatonin, the hormone responsible for sleep onset. Appetite and digestion. Serotonin influences appetite and is also found in the gastrointestinal tract, where it helps regulate digestion. Fun fact: approximately 90% of the body's serotonin is produced in the gut, not the brain. This is why serotonin plays a key role in digestive health as well as mood regulation. Acetylcholine. Function. Acetylcholine is the primary neurotransmitter responsible for muscle movement and plays a critical role in learning and memory. It is found both in the central nervous system and the peripheral nervous system. Roles. Muscle contraction. Acetylcholine is released at the junctions between neurons and muscles, triggering muscle contractions. Learning and memory. In the CNS, acetylcholine is involved in attention, learning, and memory formation. Reduced levels of acetylcholine are associated with cognitive decline in Alzheimer's disease. Parasympathetic nervous system. Acetylcholine is also involved in the parasympathetic nervous system, which controls rest and digest functions like slowing heart rate and stimulating digestion. Fun fact: botulinum toxin works by blocking the release of acetylcholine, which temporarily paralyzes muscles and reduces wrinkles. GABA. Function. GABA is the brain's primary inhibitory neurotransmitter, which means it helps calm neural activity by preventing neurons from becoming overly excited. It plays an important role in reducing anxiety, promoting relaxation, and regulating sleep. Roles. Inhibition. GABA prevents excessive neuronal firing by reducing the activity of neurons. This inhibitory effect is essential for maintaining balance in the brain's electrical activity. Anxiety regulation. Low levels of GABA are associated with increased anxiety, while medications that enhance GABA's effects are used to treat anxiety disorders and insomnia. Sleep. GABA promotes relaxation and helps initiate sleep by calming the brain's activity. Fun fact: GABA's calming effect is why many anti-anxiety medications and sleep aids work by increasing GABA activity, helping to reduce stress and promote rest. Glutamate. Function. Glutamate is the brain's primary excitatory neurotransmitter, playing a crucial role in learning and memory. It is involved in the strengthening of synapses, which is critical for the formation of long-term memories. Roles. Excitation. Glutamate increases the likelihood that a neuron will fire an action potential, making it essential for neural communication. Learning and memory. Glutamate is involved in long-term potentiation, a process that strengthens the connections between neurons and is considered the cellular basis of learning and memory. Neuroplasticity. Glutamate promotes the growth and adaptation of neural circuits, which is key to brain development and recovery from injury. Fun fact: while glutamate is essential for brain function, excessive levels can lead to a condition called excitotoxicity, which can damage or kill neurons and is implicated in conditions like stroke and neurodegenerative diseases. Summary of neurotransmitter functions. In summary, neurotransmitters are essential for everything the brain does, from controlling movement to regulating mood and forming memories. Here's a quick recap of the key neurotransmitters we've covered: dopamine, reward, motivation, and motor control. Serotonin, mood regulation, sleep, appetite, and digestion. Acetylcholine, muscle movement, learning, and memory. GABA, inhibition, anxiety reduction, and sleep. Glutamate, excitation, learning, memory, and neuroplasticity. Each of these neurotransmitters interacts with specific receptors and pathways in the brain, ensuring that the right signals are sent and received at the right time. Now let's dive into some of the finer details of neural signaling, focusing on the specific components involved in the communication between neurons, such as vesicles, the synaptic cleft, and how neurotransmitters are released and received. The synapse, the junction of communication. A synapse is the site where communication occurs between two neurons, or between a neuron and another cell type, such as a muscle cell. The synapse consists of three main parts: presynaptic neuron, the neuron that sends the signal; synaptic cleft, the small gap between the neurons, where neurotransmitters travel; postsynaptic neuron, the neuron that receives the signal. Neurons don't physically touch each other but communicate across this small gap, the synaptic cleft, via chemical messengers known as neurotransmitters. How neuronal signaling works: action potential. Communication within a neuron occurs electrically via an action potential. This is a rapid, temporary change in the electrical charge of the neuron's membrane that travels down the axon to the axon terminal. Vesicles and neurotransmitter release. At the end of the presynaptic neuron's axon are axon terminals, which contain small, bubble-like sacs called synaptic vesicles. These vesicles store neurotransmitters, which are released when an action potential arrives at the axon terminal. When the action potential reaches the axon terminal, it triggers the opening of voltage-gated calcium channels. Calcium ions flow into the terminal, signaling the synaptic vesicles to move toward the membrane of the neuron. These vesicles fuse with the presynaptic membrane and release their neurotransmitter contents into the synaptic cleft. Fun fact: synaptic vesicles are filled with neurotransmitters via specialized transporter proteins that pack them before they are released. Neurotransmitter diffusion across the synaptic cleft. Once released, neurotransmitters float across the synaptic cleft, the microscopic gap between the presynaptic and postsynaptic neurons. This space is extremely narrow, about 20–40 nanometers wide, which allows the neurotransmitters to quickly reach their target. The synaptic cleft is filled with extracellular fluid and various proteins that help regulate the process. The cleft must be cleared of neurotransmitters after each signal transmission, either through reuptake, enzyme degradation, or diffusion away from the synapse, to prevent continuous signaling. Neurotransmitter binding to receptors on the postsynaptic neuron. After crossing the synaptic cleft, neurotransmitters bind to specific receptors on the postsynaptic membrane. These receptors are protein molecules embedded in the membrane of the receiving neuron or cell. Types of receptors: ionotropic receptors. These are ligand-gated ion channels. When a neurotransmitter binds to them, they open, allowing ions to flow into or out of the neuron. This can either excite or inhibit the postsynaptic neuron by making it more or less likely to fire an action potential. Metabotropic receptors. These receptors don't directly open ion channels. Instead, when a neurotransmitter binds, it activates a G protein or another intracellular signaling cascade. This process is slower but can have long-lasting effects on the neuron, such as changing its metabolic activity or gene expression. Excitatory versus inhibitory signals. Once the neurotransmitter binds to its receptor, it either excites or inhibits the postsynaptic neuron. Excitatory neurotransmitters, such as glutamate, cause depolarization by opening sodium channels, making the neuron more likely to fire an action potential. Inhibitory neurotransmitters, such as GABA, cause hyperpolarization by opening potassium or chloride channels, making the neuron less likely to fire. The role of synaptic vesicles in neurotransmitter release. Synaptic vesicles are crucial for storing and delivering neurotransmitters. Here's a closer look at their life cycle. Vesicle docking. Before an action potential arrives, synaptic vesicles are docked near the presynaptic membrane at a region known as the active zone. Calcium influx and vesicle fusion. When calcium ions enter the presynaptic terminal due to the action potential, vesicles move to the membrane and fuse with it, releasing neurotransmitters into the synaptic cleft. Vesicle recycling. After releasing their contents, vesicles are either recycled or broken down. Recycled vesicles are refilled with neurotransmitters and prepared for future release. Fun fact: the entire process of vesicle fusion and release happens in milliseconds, allowing neurons to communicate rapidly. Neurotransmitter clearance. Resetting the synapse. After the neurotransmitters have done their job and the signal is passed on, they must be cleared from the synapse to prevent ongoing stimulation. There are three main ways neurotransmitters are cleared. Reuptake. Some neurotransmitters, like serotonin and dopamine, are reabsorbed by the presynaptic neuron through special transporter proteins. This process allows the neuron to recycle neurotransmitters for future use. Selective serotonin reuptake inhibitors. Some antidepressant medications block the reuptake of serotonin, leaving more of the neurotransmitter available in the synapse, which can improve mood. Enzymatic degradation. Enzymes in the synaptic cleft can break down neurotransmitters. For example, acetylcholine is broken down by the enzyme acetylcholinesterase into acetate and choline. Choline is then taken back up by the presynaptic neuron for reuse. Diffusion. Neurotransmitters that aren't reabsorbed or broken down may simply diffuse out of the synaptic cleft into surrounding areas, where they become inactive. Summary of fine details in neurotransmission. Neuronal communication is a complex but finely tuned process involving electrical signals, vesicle-mediated neurotransmitter release, and receptor binding. The steps include: electrical action potential reaches the axon terminal. Synaptic vesicles fuse with the membrane, releasing neurotransmitters into the synaptic cleft. Neurotransmitters bind to receptors on the postsynaptic neuron, triggering either excitation or inhibition. Neurotransmitters are cleared from the synapse via reuptake, degradation, or diffusion. This process occurs in milliseconds, allowing for rapid and precise communication between neurons, which is essential for everything from simple reflexes to complex thoughts and emotions. Brain waves, or neural oscillations, are patterns of electrical activity produced by neurons in the brain. These oscillations reflect the rhythmic firing of groups of neurons and are crucial for various brain functions, including communication between different brain regions, maintaining consciousness, and regulating sleep. Types of brain waves. Brain waves are classified into different types based on their frequency, which is measured in hertz. Each type of brain wave is associated with different states of consciousness and cognitive activities. 1. Delta waves. O characteristics, the slowest brain waves, with high amplitude and low frequency. O associated states, deep sleep. Essential for restorative sleep and physical healing. O clinical relevance, abnormalities in delta wave patterns can be associated with sleep disorders and some types of brain injuries. 2. Theta waves. O characteristics, slightly faster than delta waves, with moderate amplitude. O associated states, light sleep, relaxation, and meditation. Often seen during states of deep relaxation and light sleep. O clinical relevance, theta waves are involved in memory retrieval and are often linked to creativity and emotional processing. Excessive theta activity in awake adults might be associated with attention problems. 3. Alpha waves. O characteristics, moderate frequency and amplitude. O associated states, relaxed, calm, and attentive states, often present when a person is awake but relaxed, such as during meditation or daydreaming. O clinical relevance, alpha waves are associated with states of relaxation and a calm, alert mind. They can decrease with stress or mental workload. 4. Beta waves. O characteristics, higher frequency and lower amplitude compared to alpha waves. O associated states, active thinking, problem solving, and active concentration. Present during tasks that require focus and decision making. O clinical relevance, high beta wave activity is associated with alertness and concentration, but excessive beta waves can be linked to anxiety and stress. 5. Gamma waves. O characteristics, the fastest brain waves with the lowest amplitude. O associated states, high-level cognitive functioning, information processing, and integration. Present during activities requiring high mental effort and cognitive functioning. O clinical relevance, gamma waves are associated with cognitive processes such as memory and perception. Disruptions in gamma waves can be linked to cognitive disorders. Electroencephalogram. An electroencephalogram is a test that measures and records the electrical activity of the brain. It provides a non-invasive way to observe brain waves and assess various brain states. How EEG works. Electrodes, small electrodes, are placed on the scalp to detect electrical activity. These electrodes are usually arranged in a specific pattern to cover different regions of the brain. Signal recording. The electrodes pick up electrical impulses produced by neuron activity. These signals are amplified and recorded as a series of waves on a computer or recording device. Uses of EEG. Diagnosis. EEG is commonly used to diagnose and monitor neurological conditions, such as epilepsy, sleep disorders, and brain tumors. Research. Researchers use EEG to study brain function and understand various cognitive states, from sleep to complex cognitive processes. Neurofeedback. EEG can be used in neurofeedback therapy to help individuals learn to control brain activity patterns for improving mental health conditions like ADHD and anxiety. Clinical relevance. Seizure detection. EEG is crucial for detecting and diagnosing seizures, as it can show abnormal brain wave patterns indicative of epileptic activity. Sleep studies. EEG is used in polysomnography to evaluate sleep stages and diagnose sleep disorders. Other brain imaging techniques. In addition to EEG, several other brain imaging techniques provide detailed views of brain structure and function. 1. Magnetic resonance imaging. O function provides detailed images of brain structure using strong magnetic fields and radio waves. O applications, used to detect structural abnormalities, such as tumors, brain injuries, and developmental abnormalities. 2. Functional magnetic resonance imaging. O function measures brain activity by detecting changes in blood flow related to neural activity. O applications, used to study brain function, including cognitive processes, emotional responses, and motor activities. It's often used in research to map brain activity associated with various tasks. 3. Positron emission tomography. O function uses radioactive tracers to visualize metabolic processes and blood flow in the brain. O applications, useful for studying brain function, detecting cancer, and assessing brain disorders. It helps in understanding how brain metabolism changes in various conditions. 4. Single photon emission computed tomography. O function similar to PET but uses different tracers to assess blood flow and metabolic activity in the brain. O applications, often used for evaluating brain disorders, assessing brain function after a stroke, and diagnosing conditions like epilepsy. 5. Magnetoencephalography. O function measures the magnetic fields produced by neuronal activity. Applications, provides high temporal resolution of brain activity and is used for mapping brain function, especially in epilepsy surgery planning. Summary: brain waves reflect the rhythmic electrical activity of neurons and vary based on states of consciousness and cognitive tasks. EEG provides a direct measurement of these waves, helping diagnose neurological conditions and study brain function. Other imaging techniques like MRI, fMRI, PET, and SPECT offer detailed insights into brain structure and function, contributing to both clinical diagnoses and research. These tools and techniques collectively enhance our understanding of the brain's electrical and metabolic activities, aiding in both scientific research and clinical practice. The 12 cranial nerves. Overview and functions: the cranial nerves are 12 pairs of nerves that emerge directly from the brain, primarily from the brainstem, and are responsible for a range of sensory and motor functions in the head and neck. Each cranial nerve has specific roles, which are essential for everyday activities such as vision, facial sensation, and movement. Here's an overview of each of the 12 cranial nerves. 1. Olfactory nerve. Function: sensory, responsible for the sense of smell. Description: the olfactory nerve transmits odor information from the nasal cavity to the olfactory bulb and then to the brain for processing. Damage to this nerve can result in anosmia. 2. Optic nerve. Function: sensory, responsible for vision. Description: the optic nerve carries visual information from the retina of the eye to the visual cortex in the brain. Damage can lead to visual impairments or blindness in the affected field of vision. 3. Oculomotor nerve. Function: motor, controls most eye movements, pupil constriction, and eyelid elevation. Description: the oculomotor nerve innervates several muscles that control eye movement and regulates the size of the pupil. Dysfunction can cause drooping eyelid and double vision. 4. Trochlear nerve. Function: motor, controls the superior oblique muscle, which moves the eye downward and outward. Description: the trochlear nerve is the smallest cranial nerve and is responsible for the motor control of the superior oblique muscle. Damage can lead to difficulty in looking down and outward, resulting in double vision. 5. Trigeminal nerve. Function: both sensory and motor, provides sensation to the face and controls muscles for chewing. Description: the trigeminal nerve has 3 branches: ophthalmic, sensory, forehead, scalp, and upper eyelid. Maxillary, sensory, midface, upper teeth, and palate. Mandibular, sensory, lower face, lower teeth, and motor, muscles of mastication. Dysfunction can cause loss of sensation in the face or difficulties with chewing. Conditions like trigeminal neuralgia involve severe, recurring facial pain. 6. Abducens nerve. Function: motor, controls the lateral rectus muscle, which moves the eye outward. Description: the abducens nerve allows lateral eye movement. Damage can cause an inability to move the eye outward, resulting in the eye turning inward. 7. Facial nerve. Function: both sensory and motor, controls facial expressions, taste on the anterior 2/3 of the tongue, and some aspects of salivation and tear production. Description: the facial nerve innervates the muscles of facial expression and provides taste sensation to the front of the tongue. It also controls the lacrimal glands and some salivary glands. Dysfunction can lead to facial weakness or paralysis. 8. Vestibulocochlear nerve. Function: sensory, responsible for hearing and balance. Description: the vestibulocochlear nerve has 2 components: cochlear, hearing; vestibular, balance and spatial orientation. Dysfunction: problems with this nerve can result in hearing loss, tinnitus, and balance disorders. 9. Glossopharyngeal nerve. Function: both sensory and motor, provides taste sensation to the posterior 1/3 of the tongue, sensation to the pharynx, and helps with swallowing. Description: the glossopharyngeal nerve innervates the posterior tongue and throat, plays a role in the gag reflex, and contributes to salivation through the parotid gland. Dysfunction can affect taste and swallowing. 10. Vagus nerve. Function: both sensory and motor, controls parasympathetic functions, including heart rate, digestive tract, and sensation in the throat. Description: the vagus nerve extends from the brainstem to the abdomen, influencing heart rate, gastrointestinal motility, and vocal cord movement. It is critical for parasympathetic control of the body. Damage can affect heart rate, digestion, and voice. 11. Accessory nerve. Function: motor, controls certain neck and shoulder muscles. Description: the accessory nerve innervates the sternocleidomastoid and trapezius muscles, which are involved in head rotation and shoulder elevation. Dysfunction can lead to difficulties with shoulder movement and head rotation. 12. Hypoglossal nerve. Function: motor, controls the muscles of the tongue. Description: the hypoglossal nerve is responsible for tongue movements essential for speech and swallowing. Damage can cause difficulty with articulation and swallowing, as well as weakness or atrophy of the tongue muscles. Summary: the 12 cranial nerves are essential for a wide range of functions, from sensory perception and motor control to autonomic regulation. Understanding their roles helps in diagnosing neurological conditions and assessing the overall health of the nervous system. Each nerve has a specific function and is crucial for maintaining various sensory and motor activities, as well as autonomic functions necessary for daily living. Mental illnesses are diverse and complex, involving various alterations in brain structure and function. Here's an overview of some common mental disorders, highlighting their effects on the brain. 1. Depression. Overview: major depressive disorder is characterized by persistent sadness, loss of interest in activities, and various physical and cognitive symptoms. Brain impact: reduced activity, decreased activity in the prefrontal cortex and increased activity in the amygdala. Neurotransmitter imbalance, altered levels of serotonin, norepinephrine, and dopamine. Structural changes, potential reductions in hippocampal volume, affecting memory and emotional regulation. Clinical relevance: understanding these changes helps in developing treatments like antidepressants and psychotherapy, which aim to correct these imbalances and improve brain function. 2. Anxiety disorders. Overview: includes disorders such as generalized anxiety disorder, panic disorder, and social anxiety disorder, characterized by excessive fear and worry. Brain impact: hyperactivity, increased activity in the amygdala and reduced activity in the prefrontal cortex. Neurotransmitter imbalance, imbalances in serotonin, gamma-aminobutyric acid, and norepinephrine. Structural changes, altered connectivity between the amygdala and prefrontal cortex. Clinical relevance: treatments often involve SSRIs, SNRIs, and cognitive behavioral therapy to help modulate brain activity and improve anxiety management. 3. Schizophrenia. Overview: a severe mental disorder characterized by delusions, hallucinations, disorganized thinking, and impaired functioning. Brain impact: structural changes, enlarged ventricles, reduced gray matter volume, and abnormal connectivity in brain regions involved in cognition and perception. Neurotransmitter imbalance, dysregulation of dopamine, glutamate, and serotonin systems. Cognitive impairment, deficits in working memory, executive function, and attention. Clinical relevance: antipsychotic medications target dopamine receptors and other neurotransmitter systems to manage symptoms and improve cognitive function. 4. Bipolar disorder. Overview: characterized by extreme mood swings, including manic and depressive episodes. Brain impact: structural changes, altered volumes in the prefrontal cortex, amygdala, and hippocampus. Neurotransmitter imbalance, imbalances in serotonin, dopamine, and norepinephrine. Functional changes, abnormal activity in brain regions responsible for emotion regulation and executive function. Clinical relevance: treatments include mood stabilizers, antipsychotics, and psychotherapy to manage mood fluctuations and stabilize brain function. 5. Obsessive-compulsive disorder. Overview: characterized by persistent, intrusive thoughts and repetitive behaviors. Brain impact: hyperactivity, increased activity in the orbitofrontal cortex, caudate nucleus, and thalamus. Neurotransmitter imbalance, altered serotonin levels. Functional connectivity, disruption in the neural circuits involved in decision-making and error detection. Clinical relevance: treatment often involves selective serotonin reuptake inhibitors and cognitive behavioral therapy to target specific brain regions and correct maladaptive patterns. 6. Post-traumatic stress disorder. Overview: a disorder that can occur after experiencing or witnessing a traumatic event, characterized by flashbacks, nightmares, and severe anxiety. Brain impact: altered activity, reduced activity in the prefrontal cortex and increased activity in the amygdala. Neurotransmitter imbalance, dysregulation of norepinephrine and cortisol. Structural changes, reduced hippocampal volume, which affects memory and stress response. Clinical relevance: treatments include psychotherapy, such as exposure therapy and EMDR, and medications to address neurotransmitter imbalances and alleviate symptoms. 7. Attention deficit hyperactivity disorder. Overview: characterized by persistent inattention, hyperactivity, and impulsivity. Brain impact: structural changes, reduced volume in the prefrontal cortex, basal ganglia, and cerebellum. Functional changes: altered activity in brain regions involved in attention, executive function, and impulse control. Neurotransmitter imbalance: imbalances in dopamine and norepinephrine systems. Clinical relevance: treatments often involve stimulant medications and behavioral therapy to improve attention and executive functioning. 8. Autism spectrum disorder. Overview: a developmental disorder characterized by challenges with social interaction, communication, and repetitive behaviors. Brain impact: structural changes, abnormalities in brain growth, particularly in areas related to social behavior and communication. Functional connectivity: altered connectivity patterns in brain networks involved in social cognition and sensory processing. Neurotransmitter imbalance: potential imbalances in serotonin and other neurotransmitters. Clinical relevance: interventions include behavioral therapies, educational support, and sometimes medication to address co-occurring symptoms or challenges. 9. Eating disorders. Overview: characterized by abnormal eating behaviors and distorted body image. Brain impact: altered activity, dysregulation in brain areas involved in reward processing, such as the striatum and prefrontal cortex. Neurotransmitter imbalance: imbalances in serotonin, dopamine, and norepinephrine. Structural changes: potential changes in brain areas related to impulse control and reward. Clinical relevance: treatments often include psychotherapy and nutritional counseling, with some cases requiring medication to manage associated symptoms. 10. Schizoaffective disorder. Overview: a condition featuring symptoms of both schizophrenia and mood disorders. Brain impact: structural and functional changes, similar to those seen in schizophrenia, including abnormalities in the prefrontal cortex and limbic system. Neurotransmitter imbalance: dysregulation of dopamine, serotonin, and other neurotransmitters. Clinical relevance: treatment typically involves antipsychotic medications, mood stabilizers, and psychotherapy to address both psychotic and mood symptoms. 11. Dissociative disorders. Overview: characterized by disruptions in consciousness, memory, identity, or perception. Brain impact: altered activity, changes in brain areas associated with memory and identity, such as the hippocampus and prefrontal cortex. Neurotransmitter imbalance: potential dysregulation of stress-related neurotransmitters. Functional connectivity: disruptions in the integration of sensory and emotional experiences. Clinical relevance: treatment involves psychotherapy, particularly trauma-focused approaches, to address dissociative symptoms and integrate fragmented aspects of identity. Summary: mental illnesses affect various brain structures and functions, leading to diverse symptoms and challenges. Understanding these brain-based changes helps in developing effective treatments, which may include medication, psychotherapy, or a combination of both. Each disorder involves unique alterations in brain activity, structure, and neurotransmitter systems, emphasizing the complexity of mental health conditions and the importance of targeted, individualized approaches to treatment. Notable experiments on the brain. 1. The split brain experiments. One of the most famous experiments on the brain was conducted by Roger Sperry and Michael Gazzaniga in the 1960s. This research involved patients who had undergone a surgical procedure to sever the corpus callosum, the major neural pathway connecting the two hemispheres of the brain. This procedure was typically performed to treat severe epilepsy. The experiments aimed to understand the functions of each hemisphere by observing the effects of this disconnection. Sperry and Gazzaniga discovered that while each hemisphere had its own distinct functions, they could also operate independently. For instance, when an object was presented to the right visual field, patients could name and describe it verbally. However, when presented to the left visual field, patients could not verbally identify the object but could draw or select it with their left hand, which is controlled by the right hemisphere. This groundbreaking work demonstrated the specialized functions of the brain's hemispheres and laid the foundation for our understanding of lateralization of brain function. 2. The Phineas Gage case study. The case of Phineas Gage is a classic example of how brain injury can impact personality and behavior. In 1848, Gage, a railroad construction foreman, survived a severe accident in which a 13-pound iron rod was accidentally driven through his skull, damaging his frontal lobes. Remarkably, Gage was conscious and able to walk and talk immediately after the accident, but his personality underwent dramatic changes. He became impulsive, irresponsible, and exhibited poor judgment, contrasting sharply with his pre-accident behavior. This case provided early evidence linking the frontal lobes to personality and social behavior. Modern neuroimaging techniques have corroborated these findings, showing that damage to the frontal lobes can lead to significant changes in personality and executive function. 3. The Milgram obedience experiment. Although not directly a brain experiment, Stanley Milgram's study on obedience has significant implications for understanding brain function related to social behavior and authority. Participants were instructed to administer increasingly severe electric shocks to a learner as part of a supposed memory test. Despite hearing distressing responses from the learner, many participants continued to administer shocks under the authority of the experimenter. The experiment revealed how authority figures could influence behavior, providing insights into the brain's mechanisms for social conformity and moral decision-making. Subsequent research has explored the neural underpinnings of obedience and moral reasoning, particularly in the context of how brain regions such as the prefrontal cortex are involved in processing social and ethical decisions. 4. The Stanford Prison Experiment conducted by Philip Zimbardo. The Stanford Prison Experiment investigated the psychological effects of perceived power within a simulated prison environment. Participants were randomly assigned to be either guards or prisoners, and the study was meant to last 2 weeks but was terminated after only 6 days due to extreme and unethical behaviors exhibited by participants. The experiment demonstrated how ordinary individuals could engage in abusive behavior when placed in positions of power. Although the primary focus was on social psychology, subsequent research into the neural mechanisms behind authority, aggression, and dehumanization has explored how brain regions such as the amygdala and prefrontal cortex are involved in such behaviors. 5. The MRI studies of the brain. Magnetic resonance imaging has revolutionized brain research since its development in the 1970s. MRI uses strong magnetic fields and radio waves to create detailed images of brain structure. Functional MRI, developed in the 1990s, extends this capability by measuring changes in blood flow, thus providing insights into brain activity during different cognitive tasks. Studies using fMRI have explored a wide range of topics, including language processing, emotional regulation, and decision-making. For example, research has shown how brain areas such as Broca's area and Wernicke's area are involved in language production and comprehension. Additionally, fMRI has been used to investigate the neural basis of various mental disorders, such as depression and schizophrenia, by identifying abnormalities in brain activity and connectivity. 6. The cognitive neuroscience of memory. Research into the cognitive neuroscience of memory has provided deep insights into how the brain encodes, stores, and retrieves information. Studies involving patients with memory impairments, such as the famous case of HM, who underwent a bilateral medial temporal lobe resection to treat severe epilepsy, have been pivotal. Post-surgery, HM developed profound anterograde amnesia but retained his ability to recall events from before the surgery. This case highlighted the role of the hippocampus in forming new memories. Modern studies using techniques such as functional MRI and electroencephalography have further elucidated the neural mechanisms of memory, revealing how different brain regions, including the hippocampus, amygdala, and prefrontal cortex, are involved in various types of memory processing. 7. The Human Connectome Project. The Human Connectome Project aims to map the intricate network of connections within the human brain, providing a comprehensive understanding of brain connectivity. Utilizing advanced neuroimaging techniques, such as diffusion tensor imaging and resting state fMRI, researchers are mapping the brain's structural and functional connections. The project has provided insights into how different brain regions communicate and collaborate, revealing the brain's complex wiring and its role in various cognitive functions and behaviors. This research has implications for understanding brain disorders, as disruptions in connectivity are often associated with conditions such as autism, schizophrenia, and major depressive disorder. Summary: these experiments and studies have significantly advanced our understanding of brain function and structure, each contributing unique insights into how the brain operates and how various conditions affect its performance. From early case studies and innovative imaging techniques to complex social experiments, these investigations continue to shape our knowledge of the brain and its role in behavior, cognition, and mental health. Brain-computer interfaces, connecting minds and machines. Introduction. Brain-computer interfaces, also known as brain-machine interfaces, represent a fascinating convergence of neuroscience, engineering, and technology. These interfaces create direct pathways between the brain and external devices, enabling communication and control without traditional input methods like keyboards or touch screens. The primary focus of BCIs is to harness the brain's neural activity to operate external devices or communicate with digital systems, fundamentally transforming how we interact with technology. How BCIs work. BCIs rely on the brain's electrical activity, which can be detected using various techniques. The most common method involves electroencephalography, which measures electrical signals generated by neuronal activity on the scalp. More invasive methods include electrocorticography and intracranial electrodes, which are implanted directly into the brain or on the surface of the brain, providing more detailed data. 1. Signal acquisition. The BCI system captures brain signals that reflect neural activity. These signals are often generated in response to specific thoughts, intentions, or sensory inputs. 2. Signal processing. The captured signals are processed to decode the user's intentions. Advanced algorithms analyze the neural patterns to interpret the user's commands or cognitive states. 3. Device control. Once decoded, the interpreted signals are used to control external devices. This could involve moving a robotic arm, typing on a virtual keyboard, or operating a computer cursor. 4. Feedback mechanisms. Some BCIs include feedback systems that provide sensory or visual information to the user, helping them refine their control and improve accuracy. Applications of BCIs. 1. Medical rehabilitation. BCIs have transformative potential in medical applications. For individuals with neurological disorders such as amyotrophic lateral sclerosis or severe spinal cord injuries, BCIs can offer alternative communication methods and control over prosthetic limbs. For example, individuals can use BCIs to operate computer systems or communicate via text without physical movement. 2. Assistive technologies. BCIs are increasingly used in assistive technologies to improve the quality of life for people with disabilities. This includes controlling wheelchairs, exoskeletons, or smart home devices through thought alone, allowing users to interact with their environment more independently. 3. Neuroprosthetics. In neuroprosthetics, BCIs help control artificial limbs or sensory devices, providing users with more natural and intuitive control. This application is particularly relevant for restoring lost motor functions and sensory experiences. 4. Cognitive enhancement. BCIs are also explored for cognitive enhancement, aiming to improve memory, learning, and mental performance. This includes research into enhancing cognitive functions through targeted brain stimulation or optimizing neural activity for better cognitive outcomes. Challenges and ethical considerations. 1. Signal accuracy and reliability. One significant challenge is achieving high accuracy and reliability in signal detection and interpretation. Variability in neural signals and noise interference can affect performance and usability. 2. Invasiveness. Invasive BCIs, which require surgical implantation of electrodes, pose risks and ethical concerns related to surgery and long-term brain health. Non-invasive methods, such as EEG-based BCIs, are less risky but may offer lower resolution. 3. Privacy and security. The ability to decode and interpret brain signals raises concerns about privacy and data security. Ensuring that neural data is protected from unauthorized access and misuse is crucial. 4. Ethical implications. The development of BCIs also brings ethical questions regarding autonomy, consent, and the potential for altering cognitive or emotional states. Balancing technological advancement with ethical considerations is essential. Future directions. The field of BCIs is rapidly evolving, with ongoing research aiming to enhance the accuracy, usability, and applications of these technologies. Future advancements may include more sophisticated neural decoding algorithms, improved non-invasive techniques, and the integration of BCIs with other emerging technologies such as artificial intelligence and machine learning. Conclusion. Brain-computer interfaces represent a groundbreaking intersection of neuroscience and technology, offering innovative solutions for communication, rehabilitation, and cognitive enhancement. By harnessing the brain's intrinsic capabilities, BCIs are poised to revolutionize how we interact with the world and each other. As research progresses, the potential for BCIs to address various medical and cognitive challenges continues to expand, promising a future where technology and the human brain work seamlessly together to enhance our lives. Outro. A journey through the brain. As we reach the end of our deep dive into the intricate world of the brain, we find ourselves with a greater appreciation for the complexity and beauty of this remarkable organ. From its structural architecture to its profound influence on behavior and cognition, the brain remains one of the most captivating subjects in both science and medicine. We began by exploring the brain's anatomy, delving into its various regions and their functions, from the fundamental roles of the cerebrum and cerebellum to the nuanced interactions within the limbic system. We examined the fundamental building blocks of the brain, neurons, and their signaling mechanisms, highlighting how neurotransmitters and neural oscillations shape our thoughts, emotions, and actions. Our journey continued through groundbreaking experiments that have shaped our understanding of the brain, from the split brain studies that revealed the lateralization of brain functions to the revolutionary advances in neuroimaging that offer unprecedented views into the brain's inner workings. We also ventured into the realm of mental illnesses, shedding light on how various conditions impact brain structure and function, and how emerging treatments are being developed to address these challenges. The exploration of brain-computer interfaces brought us to the frontier of technological innovation, where the convergence of neuroscience and engineering opens up new possibilities for enhancing human capabilities and improving quality of life. BCIs represent the cutting edge of how we can interface directly with the brain, transforming communication, rehabilitation, and cognitive enhancement. As we conclude this comprehensive exploration, it's important to reflect on the interconnectedness of these topics. The brain's intricate network of structures, functions, and processes underpins every aspect of our existence, from the most basic sensory experiences to the most complex cognitive functions. Each piece of research, every technological advancement, and all of our understanding of mental health conditions contribute to a larger picture of how we understand ourselves and our capabilities. The study of the brain is not just a scientific endeavor, it's a journey into the very essence of what it means to be human. It challenges us to think deeply about the nature of consciousness, identity, and the potential for growth and change. As research continues to advance and our knowledge expands, we are continually reminded of both the extraordinary potential and the profound responsibility that comes with understanding and manipulating the brain. In closing, our exploration has only scratched the surface of what is a vast and ever-evolving field. The brain, with its boundless complexity, continues to inspire and intrigue. As we move forward, we carry with us a deeper appreciation for this remarkable organ and a renewed sense of curiosity about the future discoveries that lie ahead. The journey of understanding the brain is ongoing, and with each new insight, we move closer to unraveling the mysteries of the mind and harnessing its full potential for the betterment of humanity.